In this lecture, we're going to talk about nucleic acids. Nucleic acids might be the most important molecule on Earth because it holds the keys to the building instructions that make life. So nucleic acids, their main function, although they do have some others, is information storage. And information storage means that this molecule actually has the instructions of how to build life. So functions of nucleic acids, they're genetic material. So these little things over here that look kind of like worms, those are actually called chromosomes. Humans have 46 chromosomes. There are actually 23 varieties of them. You have 2 of each kind. When we start talking about mitosis and meiosis, we'll talk about chromosomes much more in depth. But these are units of DNA. Each one of these is a fragment of DNA, a piece of DNA that make up your genetic material. And they are found in the center of cells in an area called the nucleus. So this is where the chromosomes and the genetic information in DNA live. So genetic material, it stores information, the instructions on how to build all the components of living organisms. And we refer to units of DNA as genes. And they provide us with a blueprint for building another macromolecule, which will be the next lesson, proteins. So DNA is copied into another type of nucleic acid called RNA, which gives the instruction for building proteins out of small monomers called amino acids. So DNA allows us to build proteins. And so nucleic acids also allow us to transfer information from DNA language to RNA language to amino acid language to build proteins. And it's the blueprint for building all new cells. And it's the blueprint for building the next generation. So here, what we see is this is a cell. The nucleus is inside here. It's showing the little chromosome, this X-shaped thing. Chromosome, which is built of this DNA, which is all coiled together. And so DNA has genes that are needed to run the bodies for every day, to make you and me, to make new cells, and to make the next generation, to make babies. Nucleic acids, so some examples of them are RNA, which is what's called a single helix. So see this twisty shape here? Notice there's only one twist. That means it's a single helix. Helix is just a fancy way of saying spiral. Think spiral staircase. And DNA, which is also known as deoxyribonucleic acid. So RNA is ribonucleic acid and DNA is deoxyribonucleic acid. And it is a double helix. And you can see from the structure, so here's our double helix down here. It actually has 2 spirals. So one spiral here and the other one here. They actually intertwine. And the structure of these, they're built from small monomers called nucleotides. So many nucleotides chained together will make DNA, or if it's a single helix, RNA. Nucleotides actually consist of 3 parts. There's the nitrogen base or nitrogenous base, which just means it's a ring structure that contains the element nitrogen right there. And this is interchangeable. There are lots of different nitrogen bases that make up DNA and RNA, specifically 5, and we abbreviate them with letters A, T, C, and G, or if we're talking about RNA, U. And there's a pentose sugar, so there's actually a carbohydrate involved with nucleic acids. And this particular one is a ribose sugar, or if it's in DNA, it would be a deoxyribose sugar. And really, there's only a small, slight difference. So deoxyribose, on the second carbon here, notice we're missing the OH group that you would see in ribose. So the term deoxyribose means a ribose sugar, so this little 5-carbon guy, minus an oxygen. So D meaning without, oxy, oxygen. So ribose sugar, which would be RNA, and deoxyribose, so without one oxygen, would be DNA. And then the last component is a phosphate group. And again, this is the same old PO4 3- polyatomic ion that you would have seen in chemistry. And so because of this phosphate group, it also means that nucleotides have a charge, a negative charge. And we know that anything that is charged will also mix with water. So nucleotides are hydrophilic. Okay. So types of nucleotides. There are two types of nucleotides. And for right now, we're just going to introduce these terms. When we get into molecular genetics, we'll talk about it more. But there are two types of nucleotides. They're called purines, and then the other word down here is pyrimidines. And if you look at them, the structure is slightly different. Purines have 1, 2 nitrogenous rings, and pyrimidines have 1. So one way to remember which group is which is the phrase pure as gold. So purines are adenine and guanine. So purines, they are double ring nitrogen base, and they have adenine and guanine. And so again, the phrase is pure as gold, meaning purines are adenine and guanine, gold. Pyrimidines are single ring bases, which are cytosine and thymine. And then if we're talking about RNA instead of DNA, uracil. The other thing to think about, and this is how I remember, is that pyrimidines, longer word, smaller molecule. Purines, smaller word, bigger molecule. I don't know, it worked for me, maybe it'll work for you. So nucleic polymers, so polymers of these nucleotides, adhere together between the phosphate and the sugar. So here's one nucleotide. There's the nitrogenous base, adenine in this particular case, the sugar, the 5-carbon sugar, and the phosphate group. And then we can add to it another monomer. So here's our second monomer. We have a different nitrogenous base, cytosine, which is abbreviated with C, 5-carbon sugar, and a phosphate. So the phosphate of this nucleotide attached to the sugar of the previous nucleotide. And we can do this over and over again, and what we get is what's called a sugar-phosphate backbone. So the reason we call it sugar-phosphate backbone is just like what you see. We alternate a sugar with a phosphate, sugar with a phosphate, sugar with a phosphate, and the backbone is very strong because it's held together with all covalent bonds. So again, the sugar will bond to the phosphate, so the sugar of one nucleotide bonds to the phosphate of the next. They're covalent bonds, which means that the backbone is very strong, and this allows us to chain together millions, billions, and even trillions of nucleotides into long chains. The polymer grows in one direction, so this is going to bring into why we number our carbons and sugars. The nitrogen bases hang off the end of the sugar-phosphate backbone, so we have all of these exposed nitrogen bases. Okay, so why are the dangling bases important? Well, the reason why they're important is because if we have another chain of nucleic acids with dangling bases, they can interact together and form hydrogen bonds. So we'll see that in just a second. So we compare nucleotides from one chain of nucleotides to another. So here's one chain of nucleotides with their dangling nitrogenous bases. Here's another chain of nucleotides with their dangling nitrogenous bases. And we can make what are called base pairs. So we get hydrogen bonds between the nitrogen bases. So we get a pair between thymine and adenine, so A and T. And then we get pairing between guanine and cytosine, G and C. So purines, the double ring, so purine, there's a purine here, a purine there, will bond with pyrimidines right here. And the reason for that is if we had purine with purine, you'd end up with a DNA that would be fat at one point, skinny at the next, fat and skinny, fat and skinny. Here, because we have big molecule with little, we say consistent distance apart on each side. So the way to remember, adenine and thymine, so at, bond together, and they form two hydrogen bonds right here. And then guanine and cytosine, the two rounded letters are what are bonded together. And they make 3 hydrogen bonds. So matching bases, so A's with T's and G's with C's. What's nice about this is if you only have one side of our ladder, we automatically know what the other side is. Anytime you see a thymine, we know immediately across from it should be an adenine. If we see a guanine, across from it should be a cytosine. So the DNA molecule is not flat. It actually curves or curls around into kind of a spiral staircase. And you can kind of see, this is a 3D model that I have spinning around that you can see. But you can see there's two backbones that are intertwined. So this is two-dimensional drawing of something three-dimensional, and hopefully this animation will make you see it a little better. But we get the base pairing and we get this twisting, and we call this twist a double helix. Double because there's two strands, and helix because we have the twist. And we get the hydrogen bondings in between them. So why hydrogen bonds? Why is this important? Hydrogen bonds are not as strong as covalent bonds. You can actually think of hydrogen bonds like Velcro. They'll hold the two sides together pretty well, but if you put enough stress on the hydrogen bonds, they'll actually come apart, kind of like the two sides of Velcro. But also what's nice is you can put those hydrogen bonds back together. So you can actually open up a molecule of DNA and then put it back together. The people who discovered this double helix shape are these two gentlemen here, Watson and Crick. Crick is this gentleman up here and Watson's down here. But they actually discovered the double helix shape of DNA. Well, kind of. We'll talk about that more later. But because of these hydrogen bonds, it's really easy to copy DNA. We can actually separate the two backbones and the nitrogenous bases in the middle, and we can do what's called replication. Replication is just a fancier way of saying copying DNA. So DNA replication, also known as DNA copying, we can actually open up our DNA molecule, our two strands, and then we can add complementary bases, meaning we open it up, and if we see a thymine on the left, we can add an adenine on the right. So they match. So if you have one strand, you can build the other and build the whole thing. Why is this important? Well, before a cell divides, we need to copy the DNA so that each new cell that's created has a full and complete copy of the DNA or the instructions that it needs to run. So DNA replication, or copying DNA. So here's our original double helix, which has been highlighted in blue. And you can see here that the original double helix is split in half. And then here in orange, we're adding new complementary DNA. And what we'll end up with is two completely identical strands of DNA built from the original. And we can do this because of the pairing of the bases. So here we see that there was a cytosine on the original strand of DNA, so we add a new base complement to it, guanine. Adenine, we'll add a thymine. Cytosine, we would add guanine. Here's a guanine, we'd add a cytosine. So when does a cell copy DNA? It only copies a DNA when it's ready to divide. So in life, when we need to make new cells, so for example, if you fall and skin your knee, you have to replace the skin cells that you removed when you got the injury. And so to make new cells, we actually have to copy the DNA first. So cell reproduction. We'll learn a lot about cell reproduction later. There's actually two types of cell reproduction, one called mitosis and another one called meiosis. So in mitosis, we make an exact copy of the DNA. We give a copy of the DNA to each cell. And from one cell, we get two. So this is a picture of a single-celled organism called an amoeba, and you can actually see the amoeba splitting in half, and from one amoeba, we get two identical amoeba. And we also do this in what's called gamete production. Gamete is just a fancier science way of saying reproductive cell. So a sperm if you're a male, or egg if you are a female. And that's done by a process called meiosis. So right here you can see the male reproductive cell, or male gamete sperm. Another function of nucleic acids, which is actually a very important one, is energy transfer. And this is done with a molecule called ATP, also known as adenosine triphosphate. We talked about glucose being one of our favorite words. ATP will also become one of your favorite words. But you can see that this is also a nucleotide, sugar, nitrogenous base, but instead of one phosphate group, we have 1, 2, 3. thus the reason we call it triphosphate. 1, 2, 3,